How's it going, everyone? It's Chow here, and today we are going to talk briefly about organelles. So usually you don't have to know too much in-depth information in this particular section, so thankfully you can just go over the basics, and we're only going to go over the basics. If you need more information in regards to organelles, you can refer to your lecture notes or your textbook. But let's get started. So the first organelle that we're going to discuss is the ribosome. Now, ribosomes are kind of interesting because they're actually made of both protein and RNA components. So you obviously need to synthesize the protein components of the ribosome from a messenger RNA, whereas you have another different type of RNA polymerase that actually transcribes the rRNA section, so the ribosomal RNA section. Now, what's really fascinating perhaps about ribosomes is that you can actually find them in both prokaryotes and eukaryotes. So this is a eukaryote over here, but it's found in both prokaryotes and eukaryotes because it's not a membrane-bound organelle. And what's really also fascinating is that it actually is one of the only organelle, quote-unquote organelle, that's found in prokaryotes. Again, it's an organelle, but it's not really one that's membrane-bound, because if you think back to prokaryotes, they don't really have any membrane-bound organelles. But it is one that's found in both prokaryotes and eukaryotes, and they serve similar functions in both types of cells. Now, in eukaryotes, it's very fascinating because membrane-bound organelles are present in eukaryotes. So the ribosomes can actually attach to the endoplasmic reticulum, which is found over here. So you have these little tiny bluish dots over here, which are your ribosomes, and some of them can be free-floating, and some of them can be bound to the endoplasmic reticulum, and they have slightly different functions, but overall it's still going to be helping with translation, which we will discuss in a separate video. Now, moving on from ribosomes is kind of the control center here, the nucleus. So the nucleus, not much you really have to discuss usually in regards to an introductory course, but know that this is generally where the DNA is located in eukaryotic cells. So obviously only eukaryotes have that nucleus. In fact, that's one of the key differences between a eukaryote and a prokaryote. It's also the site of DNA replication, as well as transcription, and obviously ribosome assembly. So the assembly of the ribosome begins in this little thing here in a very dense region of the nucleus called the nucleolus. And obviously what's really fascinating about the nucleus is that it has sort of a double-layered membrane-bound envelope. And what that means is that it can kind of restrict the movement of materials into and out of the cell. There are nuclear pores that are open that allow for sort of facilitated diffusion, if I may, but these pores actually can restrict certain molecules as well. And oftentimes you have to sort of attach an importing sequence or along with certain proteins that actually bind and help mediate the process of importation, and then something similar occurs in terms of exportation as well. So yes, there are nuclear pores, but it's still very restrictive, and any diffusion across those membranes requires some kind of an aid in regards to the process. So there you go, the nucleus. Pretty straightforward, I think. The next one is a fan favorite, obviously, and that's the mitochondria. So the mitochondria is known as the powerhouse of the cell, as we came to love it so much, and it makes us all that energy that we need. It's also the site of cellular respiration, of course, which of course makes that energy that we need in the form of ATP. And what's really fascinating about the mitochondria is that it has its own DNA. And this is something called the endosymbiotic theory. And the idea, basic gist, is that in the past, the mitochondria is actually a sort of free-floating organism that became incorporated into a larger cell. And so as a result of that, it has its own DNA. What's really interesting is that its own DNA actually codes for parts of the mitochondria's machinery, But now it's become incorporated into the larger cell, so it can't just function on its own with its own DNA. It actually needs the nucleus, the nuclear DNA, in order to fully function, as well as build all those proteins necessary to allow for cellular respiration to occur. So the mitochondria is also sort of double membrane. You have the outer membrane and the inner membrane, and obviously different things can occur between the outer and inner membrane. And if you think back to the electron transport chain is actually located in the inner membrane of the mitochondria. So here they are, little sausage-looking organelles. And obviously in the center is the matrix of the mitochondria, and that is where the Krebs cycle occurs. So there you go, basic information on the mitochondria. The next thing is the chloroplast, which is sort of this plant equivalent kind of the mitochondria, although plants also have mitochondria as well. It is the site of photosynthesis for obvious reasons. So you actually have sort of thylakoids and stacked into grana that's found in the chloroplast. I don't think you have to know that, but that's just a tidbit. It's able to convert energy from sunlight into sort of chemical energy. It has that double membrane structure. It's generally considered a type of plastid, and it's found in plant cells and some protists. So there you go. It has some pretty funky things occurring for it as well. What's really interesting about the chloroplast is that it also has an electron transport chain, and it also follows the whole concept of the endosymbiotic theory. So chloroplast right here, greenish things. Cool? All right. I guess I should mention that it has chlorophyll in there as well. That might be helpful, right? So chloroplasts found generally in plants and some protists that can have the photosynthetic capability. So the next thing that we're going to mention is the peroxisome. The peroxisome is a fascinating little organelle right here in this little greenish colored thing in this particular diagram over here. The peroxisome is fascinating in the sense that it's actually something that breaks down toxic materials from metabolism. And it actually has the ability to use this enzyme called catalase that converts the hydrogen peroxide, which is kind of toxic, to water and oxygen. And it has its own special enzymes to do its own special things. So peroxisomes are very, very fascinating and very crucial to the cell, but frankly, you don't really usually have to know too much in regards to the details. The next thing is the vacuole. So the vacuole is another type of organelle that's found often in both plants and animals, but it's mostly prominent, and I guess I should say most prominent in plant cells right here. So the vacuole is a food storage area or a waste storage area, or it's a storage area for other things like digestive enzymes. It also is able to allow for support, so structural support in plants. So having the vacuole fill up completely can allow for this thing called turgor pressure to keep the plant upright. If the plant is kind of losing its water and no longer has water or other materials in the vacuole, it starts kind of dropping and drooping, and that's why if you don't water your plants, they start drooping. In protists, the vacuole is what's sometimes called a contractile vacuole, and it has the ability to sort of expel excess water. And as mentioned, the vacuole is generally larger in plants than animals. So if you look at this over here, which is a diagram for an animal cell, you can see the vacuole is fairly small, whereas in plants you often have a very large sort of central vacuole and perhaps maybe some smaller vacuoles as well. So there you go, the vacuole. Next on the list is the cell wall, which provides structural support. Some other tidbit is the fact that bacteria also have a type of cell wall, but it's not exactly the same as in plants. Generally speaking, in the scope of the course, when we talk cell wall, we're really talking about plant cells. So here's the cell wall, which adds a bit of rigidity to the cell. And it's found in plant cells and bacteria in a different type of manner, but it's not found in animal cells. So we do not have any sort of cell wall. Now, animal cells, and to a lesser extent, some plant cells as well, but animal cells are really well known for this thing over here called the extracellular matrix. And the extracellular matrix is made up of a couple of things, including collagen, which is quite important, as well as other aspects like proteoglycan, as well as linker proteins. So you can see this diagram over here. You have let's see, so we have this particular protein over here that's actually embedded in the membrane, and you have these things that jut out. So you have collagen fibers that are attached and kind of jut out. You have these proteoglycan complexes, which include, so a proteoglycan is basically sugar and protein. It's actually mostly sugar and less protein, but you have these chains that kind of jut out into the outside of the cell. And obviously, these can allow for tissue formation, as well as provide some kind of a structural support, obviously, if you have sort of a matrix on the outside of the cell. It can potentially help with movement. And finally, last but not least, it can also allow for cellular signaling. So if you need one cell to communicate with another cell, the extracellular matrix can sometimes aid in that process. So that's basically it. That's all I really have for the first section in regards to your organelles. And that's basically all that I really have for now. Now that I'm rambling, and I'm going to stop rambling, I hope you all enjoyed this short video, and stay tuned for more videos. And as always, best of luck studying, everyone.